I'm Daniel Levine, and this is the Bio Report. Body has a natural mechanism for breaking down and clearing proteins. Arvinas is among a group of companies that's seeking to harness this mechanism for therapeutic purposes. It has platform technology that enables the development of targeted protein degraders to break down disease-causing proteins. Among the benefits of this approach is that it offers a way to target so-called undruggable proteins that conventional drugs are not able to bind with in a strong enough way. We spoke to Randy Teel, Chief Business Officer and Interim Chief Financial Officer for Arvinas, about protein degradation, its evolution as a therapeutic strategy, and the range of indications the company is pursuing. Randy, thanks for joining us.
Absolutely. Thanks for having me, Danny.
We're going to talk about protein degradation, Arvinas, and its efforts to develop therapies that harness a natural mechanism for clearing disease-causing proteins from the body as a way to treat cancers and neurodegenerative diseases. How did protein degradation emerge as a therapeutic strategy?
Well, that's a good question, and it really goes back to not just the start of Arvinas, but even before that. So our founder was Craig Crews, a professor at Yale, who published a paper in 2001 that laid out the idea for the technology that we now call PROTAC degraders. And to understand how that's different than a traditional drug, probably helpful to do the quick rundown of how a traditional drug works, which is that you have what we call a small molecule or a small chemical that essentially fits into a binding site on a protein of some kind, usually, and prevents that protein from doing whatever it is that it naturally does in the body, whether that's cutting something like an enzyme, or whether that's blocking something, or adding a phosphoryl group or something like that. Essentially, the drug prevents that protein from doing whatever it was doing. Protein degradation works fundamentally differently in that it leads to the degradation and elimination of that protein from the cell that it's operating on. And Craig's initial idea was to use part of the body's own machinery to achieve that degradation. So every cell has inside of it some machinery that is involved in degrading proteins. And the cell might want to degrade a protein for any number of reasons. It might be mutated and causing problems. There might simply just be too much of it. And so the body's natural protein degradation machinery is doing that all the time in healthy cells and in unhealthy cells. And so Craig's idea was, can we somehow harness that degradation machinery that's already happening, already healthy, already working to get rid of proteins, and target it? And so really what a protein or a targeted protein degrader like a PROTAC does is it tells the cell, look, I don't particularly care what you are degrading. You can go ahead and keep doing that. But at the same time, we want you to degrade all of this specific protein that we are going to tell you, which one that is. And so in oncology and in neuroscience as well, essentially the way the technology works is one end of the PROTAC degrader binds to a target protein of interest. Let's just use estrogen receptor as an example. Estrogen receptor is a protein involved in the driving of breast cancer. And we have one end of the PROTAC that binds to estrogen receptor. The other end of it binds to that natural cellular machinery. And in so doing, it leads to the tagging or marking of the estrogen receptor for degradation, and then the body, the cell, just naturally degrades it on its own. So that's essentially how the technology works. And Craig's idea was initially achieved by using peptides or small proteins. And the big accomplishment prior to Arvinas founding was the ability to use a small molecule or a chemical to do that, which has the advantage of being a much better drug. Can be often taken orally, gets distributed well throughout the body. But that's essentially how the technology works.
And how specific are these to a target?
Well, they can be very specific. I think nearly every drug is going to have some kind of side effect. And quite often, those side effects, even however mild, are often driven by what are called off-target effects. And that's really what this question is getting to, which is how specific can they be? And what we actually found with protein degraders is that they can be more specific than the binder that they are made with, right? So I talked about a PROTAC being what we'll call a heterobifunctional molecule. It has two ends, like I described a moment ago. One end that binds to the target protein of interest, and that's the important part for now. So that binding region of the PROTAC is intended to bind to one protein, right? That's called estrogen receptor again. But because the activity of the PROTAC degrader is not dependent just on the binding, but actually facilitating that little reaction where the estrogen receptor, as the example, is tagged for degradation. There actually is a principle there where the two molecules have to come together in a certain orientation. So it's not just they bind; they have to bind in a specific orientation and way. And because that doesn't happen every time, the degradation can be very, very specific, even more specific than the original binder was. So in all of the programs that we and others create, we are essentially trying to create drugs that are as specific as possible for that target that we're interested in. And we do proteomics work, which is essentially a way to show that of all the proteins in a cell that are going down in level after treatment with a PROTAC degrader, only the target protein of interest is actually being degraded. So they are highly specific.
What advantages might protein degraders provide over other types of therapeutic approaches, and will it allow you to target otherwise undruggable targets?
That is absolutely the promise. And going back to how a PROTAC degrader is fundamentally different than traditional drugs, a traditional drug has to bind, like I mentioned, to usually some active site on a protein, which means it has to bind pretty tightly. And you have to supply enough drug and dose enough drug that all of those sites on all the protein that you want to inhibit is inhibited and covered all the time, right? But a PROTAC degrader doesn't need to do that. Because what we're not actually trying to bind tightly to the protein, all we're trying to do is drive that little reaction, that little tagging activity that the natural machinery does. We can have a PROTAC degrader that binds only weakly and transiently for a short time, for enough time for that tag to get transferred. And so because of that, we don't need to bind as tightly, and therefore some of the classically undruggable proteins might be able to be targets for a PROTAC. The undruggables are undruggable for a reason, right? Which is that we, as an industry, haven't been able to come up with a binder for that protein that binds tight enough. But if we don't need to bind as tightly, then we may be able to degrade that protein even by using a transient or weaker interaction while maintaining the specificity that I talked about a few minutes ago, because of the way that the protein and the PROTAC have to come together in a specific orientation.
Arvinas has developed platform technology it calls PROTAC. What does PROTAC do?
Well, PROTAC is really just Arvinas' name for a heterobifunctional degrader. So PROTAC stands for proteolysis targeting chimera, and really just refers to the fact that it's a chimera, a two-headed protein, heterobifunctional, that binds to that target protein and causes degradation. So while Arvinas was the first company in the space, there certainly are other companies out there that have that like the idea of protein degradation. Lots of small companies, lots of big companies out there in the space now. And really what I think Arvinas has been able to do is demonstrate really 3 times now with programs that we put into the clinic that we've been able to, in early clinical studies, right? Just being aware that none of our programs are yet approved, have made it all the way through clinical trials, that the programs that we put into the clinic have had some signals of efficacy and have been generally well tolerated. Obviously, we have to show that through the rest of the clinical trials and get that through an approval process. But Arvinas has been out in the lead in doing that. And I think we've been able to stay in the lead by continuing to show that the technology can go into new and different areas. Two easy examples that come to mind for me would be neuroscience. We're going to talk about some neuroscience degraders on the interview today. And as far as I know, Arvinas is the only company that has put a PROTAC degrader into the clinic targeted towards a neurological disease. That's LRRK2, which we just began in the clinic a few months ago. And the other thing I'd point to is that the technology is broad enough that it can really be used anywhere that cells have a degradation machinery. And what I'm getting at is we did a deal with Bayer in 2019 to start a joint venture called Earth Bio, which is actually an agricultural technology company. Plants have very healthy targeted protein degradation systems inside their cells, just as human cells do. And so we have a JV that could even be applicable in agriculture. So we've shown that the concept that Craig had invented around using the bodies or the plants, as it may be, natural machinery to degrade proteins is pretty universal and can work in a lot of different situations.
Well, take us through the process from identifying a target to identifying a lead candidate for a program. How do you go about discovering protein degradation therapies?
Right. So I'll cover this as certainly as best as I can as Arvinas Chief Business Officer, and you should certainly follow up with the real scientists who do the real work in the labs for us. But there's quite a bit of this process that is quite similar to what you'd have for a traditional drug, but then with other sort of parameters thrown in, right? So the first thing that we always do is we pick which targets we want to go after, which might seem simple, but there are ways to discover drugs, of doing screening where you essentially screen millions of potential drugs and find out what they do. We don't do that. We pick a target like estrogen receptor that we want to hit, and then we design a PROTAC to do that. And what you generally are trying to start with is, if you have any chemical matter that will bind to that protein, you start with that. Of course, there are ways and screening and other techniques you can do to try to identify chemical matter to bind a target protein of interest, but it will always be easier to start with an existing molecule if you can find that. And then essentially what we're doing is we're taking a binder to that target protein and we're turning it into a PROTAC by adding a linking region that then links it to the other region of the PROTAC that I mentioned that binds to a part of the cell's natural machinery. That part of the machinery is called an E3 ligase. So PROTAC has the two ends that I talked about. Now, once you've created that initial PROTAC, we're clearly trying to test it as quickly as we can for degradation, trying to show that we're not just binding to a target, but we're showing we can degrade it. When we pick targets like estrogen receptor, we at Arvinas try to pick targets that are not well served by inhibitors. And by that, I mean, there might be lots of situations where a PROTAC might work as well as an inhibitor. There are some great drugs out there, some great inhibitors. And it is not necessarily the case, in our opinion, that every drug needs a PROTAC. So we try to pick areas and target proteins where a PROTAC is going to have an inherent advantage. That might be because the target protein has a scaffolding function. And so it acts not just through one mechanism, but might be involved in organizing other proteins around it. So degrading it and getting rid of it will have an outsized effect. It might be resistance mechanisms. There are inhibitors that work, but then cells or cancers become resistant to them, and hitting them then with the PROTAC might have an advantage over another inhibitor. So we try to start with those targets. But once we've created the initial PROTAC, we are going through the steps that would be recognizable to most folks in drug discovery, identifying a lead, going through lead optimization to optimize that into a clinical candidate, going through all your tox studies. I think the only other thing I would say about that process that's specific to PROTACs is you have to optimize all the parameters at the same time. You're optimizing for the binding to the target protein. You're optimizing for the binding to the E3 ligase. You're optimizing for oral bioavailability if that's something that you want to have. You might be optimizing for the ability to get through the blood-brain barrier if it is a PROTAC that you expect to take into neuroscience. You've got to optimize all those things at the same time, which is complicated. A PROTAC is a bit larger than a traditional small molecule. That was a hurdle that Arvinas had to cross years ago in terms of getting a larger than typical small molecule to be orally bioavailable or cross the blood-brain barrier. So as we optimize each of those pieces of the PROTAC, you have to do it keeping in mind the entire piece. You can't optimize one end without affecting the other. But in general, it's quite similar to an overall discovery process for other drugs.
Arvinas is not alone in pursuing protein degraders. How does its approach differ from what others are doing in this area?
Well, as I said before, I think I could probably name several dozen companies in the space now that have decided to make protein degradation their reason for being. I think what Arvinas has been able to do is one, as the earliest mover in the space, we didn't have anybody to follow. And so we had to make a decision at the very start of the company of what our first targets were going to be. And the first two targets that the company picked were estrogen receptor, which I've mentioned a few times, and androgen receptor. Those are relevant in breast cancer and prostate cancer, respectively. And neither of those is what we would call an undruggable target. Both of those are very well validated targets that have been known to doctors for decades that they have a role in those cancers. But the reason that we picked those to go after first, even though, as I said before, that the ultimate promise of a PROTAC could be in undruggables and neuroscience and things like that, was that as the first company in the space, we did not want to compound the risk of a target and essentially not knowing really what would happen when you hit or degrade a target with the technology risk that we had. We were the pioneers in the space, didn't know if this technology was going to work when we started. So we figured it would be best to go after well-known, well-characterized targets first. But that's just first. So we picked those targets and have had, certainly some measure of success with them. Our lead programs are now with vepdegestrant, the ER degrader, expected to read out clinical trials, pivotal trials later this year in 2024. So we've been able to move those pretty far along. And then we have, at the same time, begun moving forward with undruggable targets. LRRK2 in neuroscience, BCL6 is one hematology target. We've done both, but I think Arvinas certainly will always be able to claim that the first few programs that we put into the clinic with a brand new technology, did something that I'm not sure that any other technology has done, which is the first few into the clinic did show some measure of tolerability, some signals of efficacy. As I said before, not through the clinic yet. We have a long way to go, but we're certainly getting closer.
Arvinas is pursuing both cancer and neurodegenerative diseases, as you've touched on, but what makes an indication a good candidate for protein degradation?
So I think it is, what I mentioned a few moments ago, which our CSO would call the tenets of PROTACs, right? It isn't just enough for us that a PROTAC would work. What we're looking for is where a protein degrader has an inherent advantage, whether that's for a scaffolding function or resistance mechanisms. I could send you a T-shirt that has 6 different measures on there for why a protein degrader fits. And to go back to the concept of comparing protein degradation to other modalities, maybe just to dive into neuroscience for a minute, right? So if we think about the existing therapies for most neuroscience or neurological disorders, generally we're talking about things like antibodies, things like gene therapies, and a PROTAC, I think, can really bring together the best of multiple worlds. So we've talked about a PROTAC being a small molecule. And because of that, we can formulate it as a tablet. It can be given orally. It gets good distribution throughout the body. It's relatively easy to manufacture. I wouldn't call it easy. I would just say relatively easy to manufacture. And that could have certainly some benefits over, let's say, an antibody in neuroscience. An antibody in neuroscience generally is delivered either IV or sub-Q. It's going to cross the blood-brain barrier at a pretty low rate. It's very difficult to get an antibody into the brain. Certainly not going to get into the cells in a brain. A PROTAC degrader, on the other hand, could be, as I said, delivered orally. It's a small molecule. We've been able to engineer them to cross the blood-brain barrier. They can get into cells, and they can degrade proteins inside cells. So I think that's a big advantage versus an antibody. Other modalities could be antisense oligonucleotides, or ASOs. Those are often dosed intrathecally with really an injection effectively into the spine. I think I, as a patient, would rather take an oral therapy than that. And again, there's advantages of how they can get into the brain for patients, which we hopefully will show with the first programs that we're putting into the clinic. So the good candidates, it's for every disease, it's going to be different. In general, we're looking for places inhibitors don't work, but we can also use the advantages over other modalities, especially in places like neuroscience, I think, to differentiate from other modalities.
You've got two cancer programs that you've partnered, one a collaboration with Pfizer to develop and commercialize a PROTAC for estrogen receptor protein degrader, and a collaboration with Novartis to develop a PROTAC for androgen receptor degrader for prostate cancer. What responsibilities does Arvinas have in these partnerships?
Yeah, they're very different. And just for listeners, the Pfizer deal was done back in 2021, and the Novartis deal was just announced just a couple months ago in 2024, and they are structured quite differently. The Pfizer deal is a global co-co or co-development, co-commercialization deal, which means that we and Pfizer share the development or all the clinical trials, submission, all those things, and the commercialization and sales of vepdegestrant. We share them equally. It's a 50-50 deal globally. And the reason we did the deal that way is vepdegestrant is our lead program, and we have had a long-term strategy to become a company that doesn't just create and invent PROTAC degraders, but we want to be a company that takes them all the way through to market, takes them all the way through to ultimately, after approval, of course, be commercializing the programs that we have created. And the deal that we did with Pfizer allows us to do that. It's a global 50-50, but we book sales in the United States and Pfizer books sales outside the United States. And that means that in support of that, we will be building out all of the regulatory and commercial infrastructure and hiring and sales force that it will take to do that. And obviously, the price of doing that is that we're paying for 50% of the development. But that was the right deal for us at that time and has obviously been critical for us as we've grown over the past few years. The Novartis deal is structured quite differently. It is an out-license of ARV-766, which is an androgen receptor degrading PROTAC, like you mentioned. And that deal was done 3 years later. And as an out-license, Arvinas does not pay for the development or commercialization. What we get in return after receiving an upfront payment is milestone payments as Novartis takes the drug through clinical trials and hopefully someday into the market. And we also get royalties downstream. And we structured that deal differently for a reason, which is that prostate cancer has some similarities to breast cancer in that there is a late line opportunity, which is, still some important unmet need for patients, going to be a smaller opportunity overall than early stage prostate cancer. But of course, to get to the early stage piece will require some trials that are long, intensive, and large. And for Arvinas, would have been quite costly at exactly the same time that we are trying to commercialize that first program, vepdegestrant, which of course is resource intensive, and is also quite likely to be at the same time when our next two programs in the clinic, which are for LRRK2 and for BCL6, are about to transition from phase 1 trials into phase 2 trials or beyond, which is exactly when they'll start to get more expensive. So we structured the deal that way with Novartis for that exact reason, which is even though it made sense to co-commercialize VEP, we didn't think that where we were as a company and expect to be in the next few years, we didn't think that made sense. So we thought it made much more sense for Novartis, who is obviously highly established, experienced, and has a good expertise in prostate cancer, to take over the development of that program. And obviously we benefit as Novartis and patients benefit.
And is the expectation in these two programs that the PROTACs would be developed as monotherapies, or would they be used in combination with other treatments?
Both. The answer is both for both therapies. So for breast cancer, vepdegestrant in collaboration with Pfizer is already, as I mentioned, in a pivotal trial. It's in a pivotal trial called VERITAC-2. That is a monotherapy trial in late-line breast cancer. But at the same time, we are also running other trials with Pfizer, with vepdegestrant, in combination with other therapies, including Pfizer's Ibrance, but also a number of others as well. So I think VEP will clearly be relevant both as a monotherapy and a combination therapy. On the prostate cancer side, similarly, what I expect Novartis's first phase 3 trial to be, although you'll obviously have to ask them for the details, what I'll predict is that will be a late line monotherapy trial for ARV-766. But I am also sure that they'll be interested in combining 766 with other agents as well. We already had, prior to doing that deal, and still have, an ongoing trial of ARV-766 in combination with abiraterone, another androgen targeting therapy. And so both of them, I think, are relevant. And I expect that to continue. It might not be true for every drug that we create, but there will be quite a few, especially in oncology, which, I think combination therapy is really becoming and is already quite a norm in some indications. There will be places where that they're used in monotherapy. There will be places that we develop them in combination as well, if that's what the situation and the indication warrants.
Well, how does Arvinas think about partnering in terms of its overall strategy? What role does it play?
Yeah, a really important part, and that's been a long-standing, statement and important piece of our strategy. We have always known that with a strategy that includes becoming a commercial stage company, that is not the same thing as saying that we can do everything by ourselves. And there are quite a number of indications out there that are really so large and resource intensive, whether that's during the development stage or even during the commercialization phase, where the resources required to be successful, not just to do the work, but do it well, require more resources than a small biotech, which Arvinas certainly still is able to manage. And I mean, look, this is true up and down, the pharma spectrum, right? The biggest companies are still partnering, the smallest companies are partnering. And so we've always said that where we believed that a partner could help us accelerate the development of a program or broaden its impact to more patients, we've always said that partnering would be important there. And so certainly as we look at our pipeline, which is pretty deep, I can definitely pick out other programs where partnering may be highly relevant in the coming years. And I can also pick out a few where I might say, I think that's one that Arvinas should be able to do on its own without needing a partner. Ultimately, we will expect to have a sales force out there that is bringing drugs and a medical team that is helping to support patients with therapies in big indications, small indications. Some will be partnered, some won't. But it's absolutely an important part of what we've done to date, not just with the Pfizer and Novartis deals. We've also done a number of deals where we were on the buy side, where we were looking to smaller companies than us to help us make sure our technology stays ahead, to look for new ways, say, to screen for ligands, to screen for what we call warheads that bind to the target proteins of interest. So partnering has been and will remain a very important part of our strategy.
On the neurodegenerative side, your lead candidate is ARV-102. It's in development for Parkinson's disease and progressive supranuclear palsy. How do these conditions relate to each other?
So both of those have at least an element that could be driven by LRRK2. LRRK2 is a kinase. It's obviously the target for ARV-102 that we're looking to degrade. LRRK2 is probably a purer driver of progressive supranuclear palsy, but is also thought to be a major driver in Parkinson's as well. So they relate to each other from the Arvinas perspective, in that they're both driven by LRRK2.
And what's the prognosis today for someone diagnosed with either of these conditions?
Well, for both of these conditions, and actually this is true for a lot of diseases in neuroscience, is that the drugs that are available now, by and large, and I'm speaking very broadly here, target symptoms rather than the disease itself. I think that's certainly true for what's available for Parkinson's and PSP, whereas doctors are able to treat symptoms but not the underlying cause. If we're able to show, and again, this is important to state that this is an early program. We just started this in the clinic. The preclinical data have been great, but it's only just begun in the clinic. If we're able to show that by degrading LRRK2, we're able to change the progression of the disease or slow the progression of either of those diseases, that's a pretty big deal, because, as I said, most drugs are attacking the symptoms. And we really think that neuroscience, because of everything that I talked about before, with respect to a PROTAC being a small molecule that could be dosed orally, that could cross the blood-brain barrier, that could get into cells and specifically degrade a disease-causing protein, we think the opportunity to bring patients PROTAC degraders could be a major game changer. I think a lot about 2019. In 2019, which is about a year after I joined Arvinas, we had our first clinical data. I think it was in October of 2019, And I'll tell you that right up to that morning, I had multiple people telling me at that conference that they just did not believe that Arvinas or anybody would ever be able to create a PROTAC degrader that could be orally available. What I'm hoping we can show with ARV-102 is that not only can we make it orally bioavailable, but it can cross the blood-brain barrier too. And I think that will be a major moment for the field and for Arvinas if we're able to do that.
Well, what's the development path forward?
Well, the development path for 102 is right now it is in a healthy volunteer phase 1 study. The neuro division is very different than the onc division for the FDA, and so and more importantly than that, just the way that you often take drugs into different areas. Neuroscience is different than oncology. The first patients that we served or attempted to serve in clinical trials in breast cancer and prostate cancer were very late line, like patients who have had a lot of therapies, who don't have a lot of treatment options, have a lot of unmet need. That's very different than if you think about a patient someday with early stage Parkinson's or Alzheimer's or something else, or any other neurological disorder. They might have a much longer lifespan than somebody with very late line oncology. So in neuro, we started in healthy volunteers. Once we've gotten through a single ascending dose and multiple ascending dose study in healthy volunteers, then we'll think about moving into perhaps patients with Parkinson's disease. But no doubt the development timelines can be quite long. One of the attractive pieces of going after a disease like PSP is that the development timeline may be a bit shorter. But in any respect, we have to show a lot here. We've got to show the technical capability of the drug. We've got to show ultimately someday that we're able to modify the disease, or at least modify some of the biomarkers that are associated with the disease. And important to point out that as we create drugs in neuroscience, it'll also be very important for the biomarkers, to come along for the ride as well, because the timelines can be so long. Neurodegenerative diseases can take quite a while, and so being able to show early on, some way or some measure that you're affecting the disease is going to be really important for us to be able to do.
Arvinas is public. It did complete a $350 million PIPE offering in 2023. We've seen a number of companies turn to PIPEs in the past year. What does that say about the capital markets today and the ability for biopharmaceutical companies to raise money?
Yeah, I was told by a banker a couple months ago that our PIPE back in November kicked off a PIPE spree. So if
You're the
anybody
one.
needs to send us, if anybody needs to send us a thank you, that they can. I think I think what is probably fair to say is that every deal is done for in a specific way for specific reasons. And for us, that structure of that deal we felt was the appropriate thing to do, given where we were. From a capital perspective, from a stock price perspective, from a what data are coming perspective, for all those reasons, we did that. We've done traditional deals, traditional capital raises in the past, and we'll probably do them again for sure. And I think the capital markets had certainly a slump, as everybody knows, that we felt like we were just starting to come out of in the first part of this year. I think that's been looking shakier, and I think the PIPE was an attractive way for quite a few companies to have a bit of a more certain outcome than they were going to get if they did a traditional raise. But I also suspect that's an idea that will also fade back in traditional raises if the capital markets become a bit friendlier, like we're all hoping that they are able to do.
How far will existing cash take you, and what's the plan for raising additional capital?
So our guidance is that the cash that we have will take us into 2027, and we are in a fortunate position with the money that we took in from Novartis for the 766 deal, plus what we had at the end of the first quarter. We've said that's pro forma cash of about 1.3 billion. So we are in a fortunate position there for sure. On the other hand, we have big plans. We've been talking about late stage, late line clinical trials and pivotal trials in breast cancer. And even though we're only paying for half, that's quite a bit. And so we certainly look at our cash balance, and we never get too comfortable, and we always have an eye out for making sure that stays a good buffer for us in the years to come. The plan for raising capital, I would say, is not too exotic. We have multiple pieces of data coming out in the coming years. whether that's pivotal data for vepdegestrant, whether that's first-in-human data for BCL6 or LRRK2, whether that's data around a potential label in the next couple of years and then getting out into launch. We think we have quite a few opportunities to go out and raise. And I think really important to point out that if everything goes to plan with the ongoing pivotal trials for vepdegestrant, as well as the pivotal trials that we have planned with Pfizer, we are really setting ourselves up to be a commercial company in the next couple of years, but then follow that up in fairly quick succession with new indications for vepdegestrant. What we're really hoping to do is start with I mentioned a late line monotherapy, but then also get into some combination therapies and approvals in those settings in late line breast cancer as well as in earlier metastatic breast cancer. So we've got a plan to get the first program launched, obviously everything going well with the trials assumed, of course, and set ourselves up where we won't be quite so dependent on the capital markets, but we're able to supplement that with revenues coming in from the first program, which obviously we will turn right back around and plow into the early stage research so that we can keep programs coming out of the research programs and into the clinic and beyond.
Randy Teel, Chief Business Officer for Arvinas. Randy, thanks for your time today.
Thank you so much, Danny. Great to be on.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.